Dear students, in this unit we will talk about the main sources of genetic variation within a population. So for a given population there are 4 different sources of variations, including formation of new alleles. Then we have altering gene number or position, then rapid reproduction, then sexual reproduction. Talking about the formation of new alleles can be arised by mutations or change in the nucleotide sequence of the DNA, which have a very significant impact on the phenotypes, and in the later generation that phenotype could be suited for life in their changing environments. While most new mutations that alter a phenotype are slightly harmful, in some cases these changes, the natural selection quickly remove such harmful alleles, so they are no more present in the population to harm their survival. Then we have altering gene number or the position of those genes, which means that either the genes are duplicated or increased or decreased in number, or maybe their position in a chromosome can be moved or shifted to a new location within the genome. Now, chromosomal changes that delete, disrupt, or rearrange many loci are normally harmful, but in rare cases they could be beneficial. Gene duplication that do not have harmful effects can persist over generations, allowing these mutations to accumulate within the population. The result is an expanded genome with new genes that may take on new function if somehow the environment changes. Then we have rapid reproduction. Mutation rate tend to be low in plants and animals, averaging about 1 mutation in every 100,000 genes per generations. But the prokaryotes and viruses, they have many more generations per unit of time, as you are aware that normally a bacteria can divide into 2 in just 20 or 15 minutes. So they have more generation per unit of time, and in these organisms, mutation can quickly generate genetic variation in their population. This helps them quickly become resistant to antibacterial in case of prokaryotic bacteria, and antiviral drugs in case of viruses. So they are quickly adapted or become resistant to the existing antibiotics. When we talk about the sexual reproduction as a source of genetic variation, in higher organisms, most of the genetic variation in a population result from the unique combination of alleles that each individual receives from its parents. So half allele comes from mother, while half alleles come from parents in a new individual. So the sexual reproduction through the process of crossing over, independent assortment of chromosome, and fertilization, it shuffles existing alleles to produce individual genotype which is different from the parents. During meiosis, homologous chromosomes, one is inherited from each parent, like one is inherited from father and the other is inherited from mother. They trade some of their allele by crossing over. These homologous chromosomes and the alleles they carry are then distributed at random into different gametes. Then because many possible mating combinations exist in a population, fertilization typically brings together gametes that have different genetic backgrounds. The combined effect of these 3 mechanisms ensures that sexual reproduction rearranges existing alleles into fresh combination, each generation providing much of the genetic variation that makes evolution possible within that population.